What's up, bio class? This is section 3 notes. Iago on the mic. We're going to talk about RNA. We'll talk about protein synthesis, transcription, translation. A lot of good stuff. Put your seatbelts on. So, by the end of this unit, you are going to learn the following. 3 main types of RNA, be able to explain the process of transcription, understand how RNA can be edited, differentiate between introns and extrons, know what a codon is versus an anticodon. It's a big one. And be able to explain the process of translation as it relates to the making of protein synthesis. So, let's start with the structure of RNA. You know at this point what DNA is. And if you guys take a look, you can obviously see we got a double helix. Right off the bat, it's single-stranded. Okay? Now, it consists of a long chain of nucleotides just like DNA does. You can see all those little vertical lines. Those are all nucleotides. One difference, the sugar group is actually ribose, not deoxyribose. So there is oxygen available. And you can see that, which is located right here. So you can see there's the OH group, sugar with oxygen, sugar without oxygen. Yes, I know there's an oxygen here. That doesn't count. But it is single-stranded, not like DNA, which is double-stranded. It does contain uracil, which you can see down here, instead of thymine. It is considered a disposable copy. So it's going to be used, it's going to then be copied and translated and then discarded of. It's not needed anymore. So that's an interesting fact about RNA. Let's move on. All right, so we got various types. You know, RNA is mainly involved in one job, and that's making proteins. What are proteins? A lot of people hear that like, oh, proteins, I got proteins in my protein shake. I got a protein granola bar. I'm taking protein powder. What does that mean? Well, proteins do most of the work in the cells. And they're kind of required, proteins are, for structure, function, and regulation of the body's tissues as well as organs. Basically, they make and regulate everything. It's the infrastructure of your body. So making proteins is a really, really important thing. And what we're going to do is we're going to use that code. We're going to use that blueprint in DNA to essentially make what proteins we want. So there are specific proteins, and when we look at that, we don't want to just remake a whole other cell. Sometimes we just want to make a cell membrane or maybe a Golgi apparatus. So that's what we can do. Now we have 3 types. We got our first one, mRNA, which stands for messenger RNA, and that takes copy DNA from the nucleus out to the cytoplasm. Numero dos, rRNA, which stands for ribosomal RNA, and that's going to help assemble proteins from amino acid sequences. It's just a ribosome. So a ribosome is actually considered RNA. Some are free-floating in the cytoplasm. Some are actually attached to the rough ER. And number 3, tRNA. tRNA is transfer RNA. It's going to be responsible for transferring each amino acid to the ribosome based off of a coded message. Now, here's the takeaway. mRNA is the only thing that's really going to have those nucleotides. If you take a look, it's right here. Everything else is just kind of excessive stuff that aren't really used in the same manner that is. So they have their own functions, and we have their own names. That's what I want you guys to know. So step 1, if you want to make a protein, let's say you want to make a cell membrane, you broke it, you got to fix it. Step 1 is transcription. And it requires an enzyme. Anything that ends in ASE, like this little puppy right here, that is RNA polymerase. And its job is to separate this. All right, so here's DNA. We're going to break it into one strand, which you see here. DNA polymerase is going to do that. Now, RNA polymerase is also responsible for finding something called the promoter region, which I have down here. Promoter region basically means that this is where I want you to start. This is a whole sequence of DNA. Notice that we didn't start here. We didn't start here. We want to start right here. And we are basically going to end right here. So sometimes we only want a certain chunk of the DNA. So we're going to unzip what we want, copy it as you can see here, and all of this is going to be done in the nucleus. So I want you to understand, this is not like mitosis. In meiosis, we are recopying the whole entire thing. We are only copying what we need. Do you have a textbook with 100 pages? You only want page 45? Find page 45. RNA polymerase is going to take you there. It's going to copy that page. and we're going to use that information to make something specific. This whole process of transcription, of copying just that section, is going to happen in the nucleus. All right. Now, this whole copying thing, you could run into some problems. Maybe you throw it on the copy machine and, you didn't put it in the right order and you basically missed a portion of it. Well, when you copy, it's kind of like writing a rough draft. That's kind of how I look at it. You're writing a rough draft. You definitely have some errors in there. If you think you write a rough draft without errors, you are wrong. But the RNA can edit itself. So you have your promoter region here. All right. The DNA contains introns as well as exons. Okay. I think I spelled that wrong in my first slide. See, I'm already making mistakes in my own slides. And introns are areas that are not involved in coding. So it's kind of like a little fail-safe. We're going to copy this whole thing, but we're only going to read and use the exons, and we're not going to worry about the introns. Okay? So that's kind of like writing a run-on sentence where you're just going on and on. We're like, yeah, we get the point. And then your English teacher edits it and says, you know what, you don't need that word, you don't need that word, you don't need that word. And we're going to cut it out. Okay? So introns and extrons are both coded, but introns are cut out. As you can see here, introns are going to be your little darker blue patches. We call that splicing. And splicing it is going to basically cut it off in your complete mature transcript. This is the good stuff. This is the best quality sentence that you wrote. And we're going to use that specific sequence to make that protein. Okay? So introns are fail-safes, they're backups, they're kind of the fat that you're going to cut off your meat before you eat it, unless you're one of those fat eaters. But either way. All right, so let's talk a little about genetic code. Amino acids are the building blocks of proteins. If you have not heard that in your life, you're not living life. And they are chained together in something called a polypeptide. Poly means many. Peptide is a bond. So if you put more than one amino acid together, and here's the wheel of amino acids in our body, we got 20, you are going to make a protein. So a protein is a polypeptide chain, more than one amino acid chained together. Now, mRNA, as I said, is a edited sequence of nucleotide bases. A's, U's, G's, and C's. This is a codon chart. And every 3 letters is considered a code. We call that a codon. This is a codon. So if I asked you a question, because you're supposed to be listening and not just writing things down, and I said, how many codons are in this line of letters? You would tell me 1, 2, 3. Codons are 3 letters at a time. We have 9 letters here. Codon, codon, codon. Now, if you take a look, we have UCG. Go here, start from the middle. U, then go to C, then go to G. Oh no, that quotes for serine. That's your first amino acid. Read the next one, CAC. C-A-C. That codes for histidine. That's our next amino acid. And our final codon is G-G-U, glycine. These 3 are considered amino acids. And they are chained together by a peptide bond, polypeptide. You just made a protein. Yay! All right. Genetic code continued. Now, we have a start codon and stop codons. Your start codon, and you'll never forget this, is meth. Yep, I said it. Methionine to be exact. And it's classified as AUG. So if you take a look, find A. Here's another code you could read too. Here's U, and here's G. AUG is MET, which stands for methionine. That is the signal to start this process. So remember we had a promoter region, right, where we know that RNA polymerase is looking for that area to copy. A start codon is an area that we're looking to go through this process of protein synthesis. We also have 3 stop codons. I have them marked here, UAA, UAG, UGA. All 3 of those let this process know when to stop. Cool beans? And we're going to do lots of practice. You're going to do a worksheet on this. You're going to do an activity on this to hopefully you really understand it. So we have transcribed. Now it's time to translate. We've got to put it in a new language. That's what translation means. So the sequence of nucleotide bases of the mRNA that you copied are going to get bumped out of the nucleus And now their location is in the cytoplasm. And they're looking for an rRNA, which is a ribosome. And that ribosome is going to read that code. So here we go. It reads its first three letters, GGC. Now, there is a tRNA out there that is going to have the anti-codon to that. So it would be CCG, which you can kind of see here. So these are tRNAs. They're going to transfer things. They're actually going to go and they're going to pick up that specific amino acid. So you read this, and that is going to match up those letters, and with it is going to come an amino acid. And as this ribosome reads right to left, bloop, 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 it's going to read those letters, get those amino acids. It's going to chain them together in a polypeptide. This is going to make a protein. Okay? So translation is basically translating, this is your mRNA, this is your tRNA, this is your amino acids. The tRNA are transfer RNA. They're going to transfer the amino acid that is specific to the code you read. Every three letters is a codon. GGC codes for arginine. UUC codes for leucine. GAA codes for lysine. GGC codes for arginine. Okay? As you saw here, they're both the same. Again, we'll do lots of practice like this. So if you're kind of like, whoa, what are you talking about, Yago? We'll get to it. All right, peeps. So here's the steps that I just talked through. I would definitely write these down in your notebook. I went through them already, so I'm not going to go into too much detail. If I were you, and I know it's extra work, I would definitely just maybe put this picture down just so you can kind of see, or maybe the picture on the previous slide. Again, we are going to have visuals in the worksheets. We're going to have visuals in the activity, and we are going to mini lesson on this. It's a skill. It can be confusing. But these are the bottom, basically the basic steps for you to understand that. So mRNA, which gets pooped out of the nucleus into cytoplasm, is attached to a ribosome, rRNA. It reads each codon, which is 3 nucleotides. Boom, 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 boom. Each 3 codes for a specific amino acid. The tRNA, which is right here, is responsible for bringing that amino acid and matching up that codon with an anticodon. So you can imagine it's going to plug in. If CGA, it's going to plug a GCU. Okay? Each amino acid brought in is attached via polypeptide bond and grows together to form a protein, which might make a cell membrane or a Golgi apparatus or whatever you want. Make muscle if you wanted it to. Last but not least, this is just kind of a larger visual just so you can see things at a bigger perspective, a little clearer. Here's your overall process simplified. So transcription, translation, DNA, copy RNA, RNA transcribed will get translated into your polypeptide chain. Here you can kind of see everything that's going on. Here's your mRNA, there's your tRNAs, it's reading the code every 3, matching up, getting your amino acids. So if you have questions, feel free to talk to your local biology teacher. Other than that, that's all I got. Look forward to a little bit of mini lesson, a little bit of activity to really help you understand this somewhat complicated process, but very, very important process. I want you to have an above average day. Yago's out of here.